next Tuesday.
Test.
Test. Okay, I know it seems to be coming faster and faster, but we're also getting closer and closer to the end of the semester. When we're talking about final drop date, which is this Friday, I talked to everybody about that in lab. So I have your grades, your estimated grade, where you stand. I'll talk to you privately. I also have your papers finished and hand them back to you for pre-editing deadline. I cannot tell you to drop. That is up to your advisor, because they know your financial situation, your schedule for your courses, for your whole career. I only know a smaller piece of the picture. I can simply tell you what grade you are likely to have at this stage in time, but you have to make the decision. And I said likely, I just say guaranteed. Okay? All right. A little bit on the background. We're going to go backwards just a little bit. On viruses, I'm going to cover these 2 points. An animal virus can be an enveloped animal virus. What does that mean? It means it has a water around the outside of the protein. An inactive phospholipid bilayer. That's called the envelope. Where does that virus get that envelope from? As it's budding out of the animal cell, it does what? Rips it right out of off of our cell membrane. So that's where he gets it. He steals it. And if you remember the movie, he even makes our cell membrane, if it's fluid, makes the H&N spikes, which are glycoproteins, attachment proteins, because the virus has to attach or it cannot infect. The movie called this glycoprotein keys, but they're called glycoproteins or attachment proteins. There are 3 places, actually, not just one, that a virus can get as envelope an inactive phospholipid membrane, if it's an envelope virus. One we just said, cell membrane, cytoplasmic membrane. Another one is, guess what? The nuclear membrane, because it flows through the nuclear membrane. It's steals an envelope from there. And what's the organelle that packages for a eukaryotic cell? Remember? Golgi body is very close, but also I'm sort of thinking about endoplasmic reticulum. Okay, Golgi body does package endoplasmic reticulum also. ER. So, summary. Virus can get its envelope from the cell membrane or cytoplasmic membrane, depending on what you call it. The nuclear membrane or nuclear envelope. And the third one is the ER, which is endoplasmic reticulum. All right? Now, another thing I'm going to show briefly. Remember we said there are RNA viruses and DNA viruses, RNA viruses and DNA viruses. Well, one thing to think of is this. I'm going to give you an example. RNA virus. RNA viruses, and this is the time when SS does not mean single-stranded. It means sense strand. In this case, SS means sense strand. And you got to know the context as to which one they're talking about. RNA virus can have a positive sense strand or it can have a negative sense strand. There are a lot of ways to teach this, and maybe there's a better way, but I try to think of metaphors to help you think, or analogs, analogies. Think of, I'm not saying it is, the positive sense strand as a messenger active RNA. As an active messenger RNA. As soon as I say that, it can code to make proteins, code to make a whole bunch of things, right? The negative sense strand, no code. Doesn't code for anything. Well, the tweak that happens here are the adjustment that viruses have to do. If you have an RNA negative sense strand virus and it has to go into its host, of course, it goes in as that negative sense strand of RNA. It can't make anything yet because it's got to go to, guess what form? The positive sense strand, which acts like messenger RNA. And then the virus can start coding for what? Having viral parts made. Does that make sense? That's one pattern. But when the viral parts are made, what do? Attachment, entry, replication or synthesis, then assembly, You have to have assembly. When you assemble them, you can't put in a positive sense strand because that's not what the virus is it? And you can't use the one that goes in, or they don't use the one that goes in. So when you do complementary strands, they make a new negative sense strand. They don't use the one that goes in. They make a new one off of the intermediate positive sense strand, and they package, guess what? They package that, and that is released out of the host cell. Out of the host cell. That's one thing to think of.
Is it still negative when it goes?
It's negative now. See, it was negative when it came in, and maybe complementary positive. Use this to make viral parts and pieces. You can't package that. That's not what he is. This is not what he is. So it has to make the complementary, and it packages the new one. Because this one's probably old, not really used. Think about it this way. If you have an RNA positive sense strand go in, the positive sense strand can simply start what? Making whatever it needs to make immediately. And often you do have to do the same thing, but we're not going to refer to that because this gets very, very, very involved. But what I do want you to know besides that one tweak is this. When you have an RNA virus, such as HIV, goes into a cell, a target, it is RNA. What did I say they as a group have? They have reverse transcriptase. That is in, what's ACE? It is an enzyme. So they're going to make DNA, and then from the DNA they make what they need. But what are they going to have to in the end package? What went? They have to make a new copy of what went in. What goes in is not what's packaged and goes out, so I'm trying to tell you. What goes in is used to control the cell, make what components are needed, do whatever type of twists and turns need to be made, but that's not what is packaged to go out. But you have to have the same thing going out that goes in, correct? So through the process, they have to make new what? An RNA base. And what is packaged? Is this packaged? No. That's not packaged because that's not what he is. The new transcript or copy is what is goes out. That's what viruses do. If you want to go a little bit deeper in than just viruses go in and take over and come out. Does that make sense? Okay. Now we're going to head for infections. And no, I don't have one. I just have frogs in my throat. All right. We're going to head for, well, infection. And you are not behaving. Very good. Thank you. Going from the focus notes, then we're going to go to a PowerPoint, and then we go back to the focus notes. This is going to be the way I'm going to do it. First, symbiotic relationships. Symbiotic relationships. You know what that means. Two organisms living together in some form. If the two organisms benefit each other, it's mutualism. And there's a gray zone about the next one. If one benefits and the other one does not, that's called commensalism. And I differ with your book. They say Streptococcus epidermidis is a commensal living on our skin. Because one of us gets benefit and the other one doesn't. It doesn't hurt. I say we both get benefit. He's a resident microbe that protects us. And he gets our shitty, well, he gets salt protection, he gets the food, he gets whatever he needs from us. But you can call it what you want. Where we tend to go to in, especially when you're talking about nursing majors, is one partner benefits X by harming the other. That's parasitism, which we also use the word pathogen. That's parasitism, also use the word pathogen. Okay? Now, our resident microbes are very, very, very important. They do what? They protect us, right? Mm-hmm. They block out other organisms that literally can't have room to land. They eat up all the food, so if another organism lands, what happens to its ability to survive? It's lesser. All right? So they can be protectors. Where do they come from? You're born. Your mother pretty much protects you in the womb. where do they come from? Mother handling you, nurses handling you, blankets in the cribs, either nursing or formula, the air that's breathed, in other words, pretty much everything that touches. So you have to acquire them, and they are very important to get as soon as you can. When an organism touches you, doesn't mean you have an infection. You can be contaminated, and, well, what? Contaminant can actually become one of your microflora and start protecting you, like it does with a baby. Or your immune system may remove it, or the resident microbes won't let it establish. But it's only an infection when it gets past your immune system and starts coming in and causing you harm. So the difference between the two. How many things do you think you accidentally eat or drink during the day that your body protects you against? Because your food's probably loaded. So you're pretty much, you get contaminated all the time. Sitting in your desk right now, you're being contaminated because I promise you the people before you left some organisms on this desk. Okay? Does that make you feel safer? Now that I've told you that, I have to tell you I lied to you. Because sometimes resident microorganisms can become opportunistic pathogens. Opportunistic pathogens. That means they take the opportunity. There are several ways this can happen. Okay? One of the biggest ones you're probably aware of is some sort of compromised or damaged or stressed immune system. In that case, what are some 5 groups who talk about this? Kids, the elderly, chemo patients, HIV, organ transplants. Okay, there are 2 other ways. Another way is right organism, wrong place. E. coli in your intestines can actually help you out. Some of those organisms give you like precursors to vitamin K. Some of them give you some precursors to folic acid. But if E. coli accidentally moves to urinary tract infection, or to urinary tract, you get a urinary tract infection. Wrong place. Right organism, wrong place. Way back when we did the bacteroides that are anaerobes in the abdomen. In the abdomen, in the intestines, no harm. Get into the abdomen, they are a bad infection. Of course, you have to have a hole, don't you? Coming from the intestines to the abdomen, that's not going to happen. Third one you know, but you're probably just not thinking of it in the way I am. What if you change the resident microbes? Change it. You have strep throat. Somebody gives you a broad spectrum antibiotic that kills what? All the bacteria in your body. But you're protected, especially if you're a lady, in the vaginal area by lactobacillus and like type of organisms that are bacteria. Well, the acidic area of that part of the body is going to no longer be acidic. It's going to start rising, and ladies often get a yeast infection with Candida albicans. So in summary, 3 ways, or at least 3 ways, as I said, there's not some others, but 3 big ways. And regular old resident can turn on you and become an opportunistic pathogen or compromised immune system. Right organism moves to the wrong place. So they have niches that they're good for you, and they move outside that niche, you're in trouble. And somebody or something changes your natural balance of your resident microflora. They're out of balance, which is the case with the Candida albicans. Does that make sense? Okay. Now, briefly, let's say we clean every single case of rabies out of the human race. There's not a single case of rabies anywhere in the human population. You can still get rabies. Why? Because there are reservoirs, animals in this case, reservoirs. Reservoirs are not, in this case, bodies of water, though it could also be that we drink from. It is a place and environment where the organism can continually come back into a population and reinfect. Rabies and raccoons can come back. You walk across this grass, I guarantee you there are clusters of tetanus spores. If you got tetanus out of the human race, it can always come back from the soil. So reservoirs are places in the world that organisms will always come back into a population, a reinfected source. You can have living and non-living reservoirs. Does that make sense? Living animals. Can't wipe out every animal on the face of the planet. That's not very feasible. And also, don't forget our nice little mosquitoes and ticks and our insects that help us out. Right? Non-living, well, water. What do you associate with water? Cholera. All that dirt has spores through it, completely and utterly. To say nothing of helping you get ringworm, which you can also get from your dog that may have laid down in the dirt. The ringworm's not a worm, it's a fungus. Gets on his hair, he comes in, you pet him, and you get, if you have abrasion, ringworm. Okay, and the next thing I'm going to go over, I'm going to come back and focus this in a minute. The organisms keep coming into us. They can, of course, make us sick if they bypass or get past the first layers of our skin and everything else. We'll go into that in a minute. There are virulence factors that help these organisms make you sick. Virulence factors. One of the toxins, which I'm going to hand out in a minute. But the other ones I want to go over right now, I'm going to go over enzymes. Enzymes. There are 4 enzymes I'm asking you to know, and I'll flip down to those and I'll come back to this in a minute. Okay? I want to do this with some pictures, which is why I'm not quite showing you some of this here. Virulence factors. I'm heading for the enzymes. Hyaluronidase, about page 14, collagenase, coagulase, and kinase. Hyaluronic acid is like a glue that's between your cells that helps your cells stay stable. Hyaluronic acid. Well, what do you think hyaluronidase breaks down? The glue between your cells. Wouldn't that enable an organism to infect further and deeper into you? Yes, it would. Collagen is one of your major proteins that make up some of your tissues. So collagenase is an enzyme that does what? Breaks it down. Breaks it down. Now the next 2, sometimes you can think of them as a set. They don't have to be a set. And you've already learned this in lab. Coagulase causes red blood cells or tissue to what? Plasma to coagulate in this case, plasma. So Staph who does this? Staph aureus does this. He will go into your tissues, form a coagulated shell around him, and hide. He's doing what with his numbers? He's increasing them. But he then wants to come out at some point in time. So he will then secrete a kinase to dissolve part of the coagulated shell around him when he comes out into your body. Does that make sense? Okay. I'm now going to do toxins. I'm going to do right now. Be sure I have this one. I'm going to start flashing. I'll give you some of the most important points first. Okay, I'm going to write down for you. Toxins, I have a handout for you next time. I can do it any number of ways. Somebody have a handout. Not about to write you on it. Toxins. Two types of toxins. Exotoxin, and some of you have handouts. If not, I'll get you one next time. They're also a pal. Endotoxin. Exo means the toxin is what? Secreted into the environment, which happens to be our cells, well, our body. Endotoxin is a structural part of the bacterial cell. There are 3 types of exotoxins. Neurotoxins, guess what they get? Okay? What does the word entero mean? That means often hits the intestines. That's where polio starts. Doesn't finish there, but that's where it starts. And just flat out break you down. Cytotoxic, which means it destroys cells. Destroys cells. That would be gangrene. Doesn't it just dissolve your cells? Pretty much it does. These tend to be proteinaceous. They tend to be very powerful. Neurotoxins are certainly powerful. Remember, Clostridium botulinum is one of the most powerful neurotoxins there is. Our body tends to fight them with antibodies, which we'll go over to into the next chapter, antibodies. Often think of an antibody as shaped like a small Y, but that's the simplest one. Well, what do we say viruses have to do to make us sick? They have to attach. Toxins to have an effect have to attach. Everything is about matching patterns on the invader and the target. But if you cover that toxin, which is a small protein in most cases, and cover it up with antibodies, that's like wrapping it in duct tape, can it dock or attach to your cells? No. So that's one reason you have toxoids, which are actually vaccinations which are made from antibodies. And they had to get those antibodies by injecting an animal with what? The disease, to get the antibodies made, which they then collect from the plasma to give to us. Does that make sense?
What was the example you said for cytotoxins?
Cytotoxins, gangrene. 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 Okay? Now, endotoxins, you know this. There's a bacterial cell with 2 cell membranes. And with its head buried in the outer cell membrane is a lipopolysaccharide. The polysaccharide tail goes out into the environment, our body. The head is lipid A. But lipid A is buried in the outer cell membrane. Lipid A is another word for structure. Lipid A is endotoxin. Endotoxin is lipid A. One is simply calling the structural component in the outer cell membrane. The others, they're talking about more of the effects. I said 2 cell membranes, ground positive or ground negatives. Ground negatives. And what do we say happens? Well, the cells are left alone, assuming they're not killing with an infection. The lipid A is not released into your body. But if you crack open all these cells with a massive antibiotic, and there's like 10 million of them, you're releasing what in massive quantities into your body? Lipid A, which is endotoxin. And then we get those effects we talked about. Bleeding, shock, hemorrhaging, bleeding, all the way, if it goes all the way to anaphylactic shock. So those are the 2 parts. This one is secreted. This one is a structural part. Structural part. Be able to know the difference in the 3 types of exotoxins. Now, staph, think about what I'm about to say. Staph has a heat-stable toxin. Heat-stable. So, unless you cook it really hot, what happens to that toxin if you think you're going to kill it off when cooking for your dinner? It's going to make it through the heat in many cases. So we're talking about, we had enzymes, we had hyaluronidase, collagenase, coagulase, kinase. 2 of them tend to be a pair with Staph aureus. That's one set of virulence factors. Then we have toxins, which organisms can secrete. We talked about that, exo and endotoxins. So those are some of your virulence factors. Don't focus on virulence factors. Adhesion factors, we're going to go back to the part of your focus notes for adhesion factors, because this came from your book. And your book happened to do it in this pathway. Coming back to page 9. What I say it has to do if it's going to affect you. It's got to attach. A ligand is something that helps an infective agent attach. Now, this is just terminology. In viruses, the glycoproteins are called attachment proteins, and the patterns match. You get attachment, the virus attaches, then it enters. Okay? For bacteria, you still have attachment proteins, but they call them a different word. They call them adhesins. Adhesins. And here it is right here. For bacteria, I'm sorry, thank you for spelling it out. Thank you for spelling it. Back on page 9, y'all tell me. Back here on page 9, adhesins for bacteria, and for viruses, attachment proteins. But I was already calling them attachment proteins, which are your glycoproteins. So how do they make you sick? Is next virulence factors such as enzymes, toxins, and they have to have the right ability to attach. You have a terminology sort of between viruses, attachment proteins, bacteria, adhesins. All of them as a group are called ligands. Okay? They can't attach, they can't make you sick. Neisseria gonorrhea, for instance, has the adhesins on its fimbriae. If it loses its fimbriae, it can't attach to your reproductive cells. It can't give you gonorrhea. It can't give you gonorrhea. That makes sense. So 3 big ones right now. You know, 3 big pictures. Toxins, enzymes, and the ability to attach. Attachment. Does that make sense? Okay. Now, I'm going to go from beginning to end because I want to pull those together since your book separated them out. Symbiotic relationships we already talked about. I have a picture for that for later. Talk about resident microbiota and where they come from. I'm not making you learn this page right here because every single person is slightly different. The organisms that are in this person's intestine are not the same organisms in that person's intestine. In fact, they're doing fecal transplants from people with C. diff, well, people are healthy, into people who have C. diff. Because what? The organisms are different. Organisms are different. And they're thinking about making pills. They're thinking about making pills now. They already have with healthy microorganisms in them and put those into a person's intestine. Try to get the right ones. They're even going so far to say that some people might be more overweight than others because of the type of bacteria in the intestines. Now, is that true or not? We don't know if that's true or not. Not yet. Not yet. So this can actually vary quite a bit. We talked about that if you have an organism contaminate you, that doesn't mean you're going to have an infection. We talked about the 3 ways that resident microflora can become opportunistic pathogens, right here. Okay? So, move to unusual site and compromised immune system, or somebody unbalances or something unbalances your resident microflora. Right there. Okay? So we did that. Reservoirs, animals. Reservoirs are what? You can bring something from the environment that reinfects the population. Animals, it's a little bit more dangerous when wild animals mix with domestic animals. Because the wild animals are not vaccinated, not given all those shots. Your pet goes out, mixes up with a wild animal, or walks through the yard where that wild animal had feces and urine and shedded whatever, comes back into your what? House. A term for wild animal is sylvan, S-Y-L-V-A-N. That's one of the terms. Okay. But that mixes it up. Or to give you a picture, this isn't an infection for us. A vet once told me, because a person had mice, ear mice that kept showing up on the dog. And they kept clearing it, the dog kept getting ear mice. They kept clearing it, the dog kept getting ear mice. So, what happens was there was a feral cat colony. We're getting started on feral cat colonies for being disease sources. Alright, I like cats. I just don't like disease sources. Cats walk back and forth through the what? I said animals don't have to actually move. Through the backyard. Drop whatever they're going to drop. Ticks, your urine, feces, eggs in their feces for tapeworms, mites. So every time the dog goes into the backyard to the bathroom or play for a while, it picks up ear mites. And that vet, whether he's right or wrong, because you always have to check information, said as long as there's a feral cat calling in the area, you will never, ever get rid of ear mites in your own pets in your own house. Okay? And another vet may differ. I'm not going to say that's written in stone. But it gets worse than that. What if we're talking tapeworm eggs? What if we're talking roundworm eggs? Okay? You know what a zoonosis is? Jumps from animals to people. Now, how do you get zoonosis? Scratched, bitten, okay? Get some sort of animal feces. Kids are bad for playing in the yard, putting their hands down. They don't know they touched anything because it's mostly degraded. Touch their mouths. Don't cook your meat well enough. There's no help with problems. And who's going to help you out? Mosquitoes. Mosquitoes will always help you out. Ticks too. Ticks. One person, was it this class that told me went to the women's bathroom and they found a tick that was on the floor. It already fed and was swollen. Yeah. That's not good. Okay? That's just not good. Your puppy dog, and I like dogs too, can be a reservoir. Helminthe, know this right now, means worms. Means worms, but I'll probably help with that. Can be a tapeworm reservoir. Okay? It just is. So treat them for worms. Roundworms too. That's ringworm is not a worm, it's a fungus. You get that from the hair of any fuzzy animal that you love. If there's an abrasion or a slight cut. There are human carriers. I've got some pictures to show you. That means a person who has no symptoms. And they probably won't get symptoms. And in some cases they will. Typhoid Mary is one. I'm going to mention her in my, I'm going to give you a PowerPoint after this with some pictures so it's not so dry. You can have strep carriers. You can have MRSA carriers. And the one in your book was a gentleman that was at a construction site. He was urinating not in the porta potty, but in a culvert along and got everybody else sick. So, yeah, what can you say? People can be carriers and they have no symptoms. Non-living reservoirs, we talked about water, okay? And of course dirt, and actually some others. There's the ligands, which I went and combined with the virulence factors. Okay, that's still your adhesives. Okay, we already said, this is a brief point, most of this you already know. A sign versus a symptom, and most of this I'm leaving to nursing, because I'm teaching microbiology on nursing. Alright? Sign means what? You can see the person throw up. You can see the diarrhea. Symptom, at least the way your book says it, if nursing changes it, though I know, means the person says, I am nauseous. You can't see the nausea. You can't see them say, I hurt inside, my abdomen hurts. Like the little girl did when her mother gave her tapeworm pills. Okay? That's just a slight thing.
How about swelling and
That would be sign. You can see it. You can see it. But I think that's a fine-tuned definition of difference. I teach it because it's in your book. I'll let nursing take care of that. Because they'll have probably better definitions for it. This is my book. Koch's postulates, you remember that. Isolate the organism from the sick animal. Have to grow it in pure culture in the lab. Reinoculate a sick, healthy animal. It gets the same illness, it gets sick. Isolate it back out. That's supposed to group organism A causes disease A. You can't do that with people. Well, you probably even do it with animals to a certain extent. But you can do epidemiology, which is to study the data. You already know that Haemophilus influenza was misnamed. Why is it misnamed? Flu is a virus. Haemophilus influenza is a bacterium. The name is stuck. Now they can rename Streptococcus pneumococcus, but they can't seem to rename Haemophilus influenza to something better. It was a secondary what? Bacterial infection from people that had the flu. And they couldn't see viruses then. They couldn't see them at all. No natural spreads. Okay. Couple things. This is more into the level of correlation. You gotta be careful of correlation because it can be utterly wrong. like storks bring babies. Actually, storks were going to the chimneys, which were warm and cold areas, and if a person was rich enough to have a house in a chimney, they tended to be a family. Families tended to have children. Watch your correlations. They can be completely wrong. So when I'm telling you this, I'm still telling you it's a correlation, not fact. Well, sometimes they say if you combine two organisms, they're saying right now if you have hepatitis B and hepatitis D, that people tend to get cancer. Now, that's an observation, not a known outcome. Hepatitis C, but two different hepatitis would give you cancer. Don't forget combination. Ethics we talked about. Okay. They still don't know Parkinson's disease, Alzheimer's, multiple sclerosis. I've heard everything. Some people say there's an association with Clostridium perfringens with Parkinson's. Some say there's an association with aluminum with Alzheimer's. So, get the idea? They're still what? They're still learning. Still learning. If they do learn, let me know. And I hope we all know it before you hear that. Virulence we talked about. We did adhesion, toxins, extracellular. We're going to go. Aren't you tired of hearing about biofilms?
Yeah.
Okay, fair enough. Biofilm, what is it?
It's a collection of
Association of different organisms. They arrange themselves into oxygen characteristics. They collect nutrients. They're resistant to antibiotics. Kill them off, they grow back. Kill them off, they grow back. CDC is saying that they're more and more of a problem than they thought, especially with catheters. Catheters are bad. They're highway systems into your body. Highway systems. Who's using it right now? Gram-negative, antibiotic-resistant, new wave of bacteria. The CREs. CREs. I hope y'all don't see them. Hope you don't see them. They're your enzymes, hyaluronidase, collagenase, kinase. If something has keratinase, just back you up. What's keratin? Fingernails, skin, hair. Oh, what's the fungus going to do? It's going to eat your hair. Fungal infection right around that cuticle is hard to get rid of. It is hard to get rid of. You have to put an internal organism. When they do, they break it down and eat it. Once they get to the wetter parts, they move better. And entamoeba has a mucinase. All that means is entamoeba, who's normally not harmful to you, opportunistic action, can eat through the mucosal wall of your intestine under certain immune-compromised positions. But you already have those threads of information. This is what you heard. Normal resident, immune-compromised. Normally doesn't hurt you, but he can, if he has this enzyme, drill a hole through. All that ties in what you already know. You already know that. Okay? Toxins we did, endotoxins. This is very important. Anti-phagocytic factors. And you already know the first part. Are you starting to realize that as we're building knowledge, you're actually coming back to what you already learned, what you already learned, what you already learned? Capsules. That's a capsule. Not protein. Polysaccharide watery layer on the outside of the cell wall. Some organisms have them, some don't. Some can make them, some may not make them. Remember Streptococcus pneumonia? With a capsule, it can do what? Give you pneumonia. Without a capsule, it cannot give you pneumonia. Okay, what were the three things I said about capsules? We're coming back to where we were before. Think of circles. It can hide. And will hide the organism because a capsule is regular, generic, everyday type of sugar, polysaccharide. Immune cell may just go right by, see the cell. Two, can help start that biofilm. So we're back to biofilms. All the knowledge starts interlinking, and it keeps interlinking. Helps start to stick, to attach, start the biofilm. Third one, get away. It's what? It's like a wet bar of soap. You try, the immune cell tries to grab it, and it just slides off. class out. And next unit, we'll learn how the immune system sticks a hook on it so it can grab it. Because there's two ways it can do that. All right, antiphagocytic chemicals, so one's capsules, the other ones are chemicals. Lysosomes. Neisseria. Let's talk about phagocytosis first. Phagocytosis. What does that mean? Your immune cell is eating the invader. We said viruses want that to happen because some of them use that as a way to get in. But usually, it protects us. Okay? So you have some sort of neutrophil or macrophage, goes up, surrounds the bacterium. The bacterium is now in a little vacuole called a phagosome because you just had phagocytosis. That's your phagosome. Floating around inside the immune cell. Coming off of the Golgi body are little teeny lysosomes. And remember back, I said the lysosomes are loaded with three things. Think, tying back to metabolism. Toxic oxygen byproducts, acidic pH, okay, and breakdown enzymes like proteases. So if you have a lysosome floating here, and you have a phagosome floating here, this has kill components, this has the captured invader. What happens if you take, we already talked about this, two phospholipid membranes that get close together, they fuse. So what are you dumping on the invader? Toxic oxygen byproducts, acidic pH, and breakdown enzymes, proteases, lipases. What should happen to that invader? It should be chewed apart. By the way, they combined the name. They now call it a phagolysosome. Phagolysosome. Neisseria chews. He chews very well. He won't let the lysosome fuse with the phagosome he's in. He won't let the lysosome not that. American know-how. Tap it, kick it, hit it. Okay? He will not let the lysosome fuse. So if the lysosome doesn't dump its toxic products onto the Neisseria, what happens? The Neisseria lives. It lives. Okay, we already talked about fimbriae, which is strep pyogenes, and an M protein is also on strep. Think of the M protein more for strep. Strep tend to have an M protein. It helps them resist phagocytosis, which means the immune cell is not able to eat them very well. Can't eat them, it can't break them down. Last one's very simple. Some bacteria release perhaps toxins or something else. Flat out kill the organism coming, well, the immune cell coming out. Flat out kill the phagocyte, no, the macrophage or the neutrophil. Flat out kill the macrophage or the neutrophil. The bacteria kill. Third one, destroy the attacking phagocytic white blood cells right there. And yes, there are phagocytic which are the macrophages and neutrophils. Just kill it. And the bacteria then toodles on and does what it wants. Does that make sense? Okay, stages of infection. So we have another time to get the handout. Modes of transmission. Modes of transmission. Contact. Some sort of touch. Okay? Warts are highly contagious. Kissing, touching, being scratched or bitten by an animal. Okay? That will happen. I have some pictures for this as well. I'm going through this. Indirect contact. Well, people sharing needles. Sharing glasses. That's how the breakout occurred of mononucleosis that broke out in the local schools. Money. Y'all always handle other people's money. That's been through what? Other people's hands. Okay, a fomite, know that term, F-O-M-I-T-E, is any inanimate object that carries bacteria. If a person takes those bed sheets and does this, or even just jerks on them, the person's, skin has been shaved. Those skin cells are now thrown up into the air. and the fomite can be breathed in. It can make the next person sick. Yeah.
You said that's any object that carries what now?
A pathogen, a bacteria, or could be a virus. Droplet and airborne transmission, they tend to say sort of gray zone. Okay, airborne if the droplet goes more than a meter, droplet if it's less. Vehicle, that means your nice little drinking water, body fluids, foods. Okay, here's your airborne, which is more than 1 meter. Remember the sneeze? Don't you think that sneeze is going to spread everything through the whole room? AC units are great for Legionella. It's spreading air. It's spreading everything through the whole building. Sweeping is terrible. Sweeping is putting everything up into the air. Okay? Hantavirus loves to be in dust. It's in mice feces. More of a problem where it's out drying in the west and deserts. Less of a problem here. Because the mouse species, which have the hantavirus, which is a respiratory virus, dry up, they turn to dust. Breeze comes through or somebody sweeps. What goes up into the air? Dust. In this case, dust is the fomite. Dust is the fomite. Waterborne, fecal to oral route is the major source of disease for this world because most of our water has what in it? Feces. Especially people that don't live in a rich area. The water gets contaminated. So that's what's called. Food, don't cook it well enough. Worm eggs, definitely. Okay? Somebody who didn't wash their hands. I've seen people at restaurants with runny noses just running, serving people food. Haven't y'all seen that too? Is that you in it? I have. Okay. Parasitic worms, any teeny little egg that gets in there. Any fecal contamination, which we're going to do the coliforms later. And E. coli in your hamburger. Okay, body fluids, you know that. You have to protect yourselves. Urine, pus, vomit, blood, you know that. Vector means the bugs. The bugs. In this case, you can have biological or mechanical vectors. Mosquito is a biological vector because while he's flying to hunt you down to bite you, he's increasing the number of the plasmodium parasites inside himself. He's increasing the number, they're replicating inside him. So by the time he bites you, he's saving more to give you. Mechanical vector simply steps on something or its wings brush against something, and it simply mechanically transports it to the next place. To the next place. It's mechanical. Okay? If the little flies, have you seen these green flies? If they step, these they go around dog food a lot of food. If they step on the poop and they come into your house and they walk across your sandwich, they have mechanically transferred the organisms. It's people that like y'all weren't sure what that meant, so I gave it to you. Okay. You know this from nursing. Acute, sharp symptoms, very intense symptoms. Chronic, goes on and on and on and on, doesn't give up. Latent can be triggered like shingles. We talked about latency. I'm not going to go too much into the others, except contagious means it can carry over. Non-communicable, excuse my English, is a cavity. Can you catch a cavity from somebody else? No, you can't catch a cavity from somebody else. Epidemiology, and we'll go back to the handouts in just a minute. Where something starts, how it spreads, overlaps with infection control, how to stop it. And here's your word you've heard from me 1,000 times, endemic. What? Always present, sporadic. It pops up here, pops up there. They don't exactly know where it's going to be. Epidemic, the whole country or the whole state. Pandemic was 1918. What does that mean? The entire world. And an H1N1 didn't do anything. We didn't really have a pandemic there. CDC, y'all know you have reportable diseases. You have to report. AIDS. Why would you report AIDS? Because it's harming so many people. I wish I could put the answers here. We'll have to change this in the future. Anthrax, because it's a potential terrorism. Botulism, because something went wrong with the what? Food industry. Brucellosis a spontaneous abortion in cattle because it can harm the entire economic cattle industry. Cholera because it means breakdown in the what system? Water system. They usually have a reason for what they're worried about, on what they're worried about. I'm not going to worry about incidence versus prevalence. Nosocomial infection, please tell me what that means.
You get it from the hospital.
You get it from the hospital. Person went in sick and came, when they went in well and came out sick. Went in with one illness and came out with another. Went in for surgery, kind of like this horrible movie I have, died of an infection that they call in the hospital. Well, think about it. What's in the hospital? Every sickness you can think of. Let's take everybody, put them in the same place and close the doors. Okay, go. But what's the alternative? You've got to have the doctors and the specialized equipment. You can't afford the specialized equipment in 100 different places because it's too expensive. It's a balance. It's one of the balances. Okay. Now what I'm going to do is I'm going to go over a couple more things. Yeah, she's got to go over more things. She does that for you. I want to go over portals of entry. I have a handout, don't worry. And first line of defense, so there's going to be some rules. Portal of entry and first line of defense. We're going to do first line of defense. Who needs this one? I don't have plenty of them. First line of defense says skin, and we just do it resident microbiota. Skin, resident microflora. It says first line of defense. First line of defense. You don't have it on here. We're not trying to say you can't have it here. First line of defense.
Oh, I did take a look.
Okay. I'm going to read you for this. Our Xerox machine is in a mood. That's why you have the black glowing. Okay? That's the best you can give us right now. Another one? Anyone else? Well, okay. First line of defense. Well, obviously, your skin. Your skin. Very, 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 very important. One is a physical barrier, right? It physically stops anything from getting in short of being cut. Short of being cut. Skin is also, you know how it works. You have living skin cells. The dermal layer. They rise up to the top. They dry out and they die. And then the top layer of the skin is constantly shedding. It also tends to be dry unless you're sweating. It also tends to be salty because of the sweat evaporating. You also have sebaceous glands or sebum. That is where the skin does this partially to make it acidic and partially to keep your skin flexible. So we have acidic, we have salty, we have dry, we have a constant shedding, and that's just the beginning. That's a pretty good system, right? The organism may attach, but he may be shed at all. He may not be able to take the acid. He may not be able to take the salt. He may need more water. All of those are very important. Very, very important. You also secrete a few, or not a few, but a lot of antimicrobial compounds. The one right here says defensins under number 1. That's just defense with INS on it. Defensins. The next one down, number 2, is dermacidins. DERMCIDINS. They kill, both gram positives and gram negatives. We talked about salt. The next one is important, but you heard about it with viruses. Lysozyme. That's number 4. Lysozyme breaks down only one compound. Breaks down. Peptidoglycan. What's the target? Bacteria. Don't worry about this sentence on the bottom is cut off. We're not going to worry about that. Resident microbes, we already talked about them. They're right here. You get some vitamins, you get some folic acids, precursors to C. They did do, that top section's about an experiment. They took animals young ones, raised them with quote-unquote no bacteria, everyone developed an autoimmune disease. Every animal that was raised without any bacteria around it developed an autoimmune disease. Mucous membranes, they tend to be much more wet, much wetter, but mucus still does what? It still sloughs off, still sheds. Some case you have some small hairs to help sweep things out. Underneath all of these mucous membranes and the skin and everywhere, you have dendritic cells. D-E-N, dendritic, D-R-I-Tic, T-I-C. They are not the dendrites in your brain. Okay, that's something else. Those are still in your brain. These are immune cells that you might want to think of them like amoeba. They get underneath every boundary between you and the environment. They send out little pseudopods. What do phagocytic cells do? They engulf the target, phagocytosis or endocytosis, pull it in a phagosome. Eventually they're going to break it down, like using lysosome, but they do something extra. They run attack death. They get that first invader at the surface of the border of your nation or your body with the whole rest of the world. Don't you want a scout out there? They get the first invaders, run to the lymph nodes, on the way of breaking the invader down into pieces, present the pieces in the lymph nodes to the next immune cell to get the next adaptive system to start attacking. So that takes weeks. Okay, and there's your dendritic cells down here at the bottom. So don't forget them. Of course, almost all your secretions have lysozyme. On the back, I'm going to talk about iron ores. It says second line of defense, it says chemicals. This case, I'm going to first talk about iron ores. Fe is the periodic chart symbol for iron. Fe is simply the periodic chart symbol for iron. In this case, some bacteria need iron to infect you further. And if they get iron, they do a better job of infecting you. Okay, that's what I do. Over here, iron is needed. Well, we need it for oxygen. It helps the red blood cells hold oxygen, hemoglobin. Staphylococcus. Let's get some points. Transferrin is a protein that our body uses to move iron around. That's just the name of it, transferrin, because it does what? It transfers iron. Does that make sense? Down over here, Staphylococcus will have citiophores because it's stealing the iron from our bodies. It will take the iron if it can from our plasma. Our body then makes lactoferrin, which will steal the iron back from the citiophore of the Staphylococcus. Get it? It's like you put a little ball out there, and everybody's fighting over who can get it better. Staph then gets mad, and it goes over to this part right here, and it makes a hemolysin. What does that mean? Lysis, red blood cells. It just simply does what? Cracks open the red blood cells. How much iron is in there? Let's go over it again. Transferrin is something that your body uses to transfer iron. Okay? It's a carrier. Think of a little boat. It's not really a boat. Staph cheats. He puts a little citiophore that has a higher affinity for iron than little boats. He steals the iron from your blood, your bloodstream, not your blood cells. Then your body makes lactoferrin to try to steal the iron back. And then Staph takes the final step, produces hemolysin, and just does what? Cracks open your red blood cells and says, I'm taking your iron no matter what. An infection. That's not sort of nice, is it? Now, Neisseria cheats. I said transferrin is what your body uses to help transfer iron, or something, one of the points that your body uses to transfer iron. Neisseria simply makes transferrin ports or receptors on himself. So the iron goes straight to the bacterium. He simply makes a mimic. He simply says, well, if you're using transfer iron, I'm going to put some on my cell wall and you're going to give it to me. That's pretty smart, isn't it? That's iron wars, and FE stands for iron. All it says over there with the immune defense cells is some definitions. 2 words to know there are margination and diapetesis. Margination and diapetesis. In margination, what's happening is your immune cell is rolling along. It's going through your blood vessel. There's some sort of chemical signal that is released onto the surface of the blood vessel over there. Soon as your phagocyte, which is phagocytic cell, which is macrophages and neutrophils, gets that chemical signal, it stops. Stops in the garden hose. It's like it's going through the garden hose and it stops because there's a signal. The signal's telling that there's a what deeper in the tissue inside, an infection. So margination is the phagocytic immune cell, i.e. macrophage or neutrophil, moving along, sees the chemical signal, stops, margination. Diapetesis is when it squeezes through, it meaning the immune cell, squeezes through the sides or between the cells of the blood vessel and is heading where? Into the tissue where the what is? Infection. It's got to get to the infection to fight, doesn't it? So diapetesis is the actual squeezing through of the phagocytic immune cell, macrophage or neutrophil. To squeeze through between the walls of the blood vessel to go into the tissue after the infection. It's also called positive chemotaxis because it's heading what? Toward the signal. Toward the signal. Does that make sense? Does that make sense? Okay. So, that's pretty much that. We've done this. Which one is? Now, I want to get a little bit into the next unit, and then I'm going to do a review of this unit we just covered, so I won't leave you completely alone.